Okay, good morning everyone. Welcome to today's Progress Report. Our first speaker is Jose Maria Gonzalez from Structural Computational Biology Group. Chema obtained his master's degree in bioinformatics and computational biology at Universidad Complutense de Madrid in June 2004. And after that he was working in the area of proteomics and molecular imaging in different laboratories in Spain and also at the University College London in England. And in March 2006 he joined CNIO as a research assistant in the laboratory of Professor Alfonso Valencia. And today he will talk about mapping and interpreting disease-associated mutations within the framework of human protein kinases.
Thanks, Eric. So, genome analysis projects provide a huge amount of mutations that are not always associated with disease. Associating a disease with a mutation or a mutation with disease it requires a lot of effort, time, and money sometimes. So we propose here the use of computational biology techniques to extract the features common to all the disease-associated mutations and try to build a system which is capable of predict the pathogenicity of a mutation, or at least to prioritize the mutations that are more prone to be disease-associated so they can be analyzed later on in the wet lab laboratory. This is not definitely a novel idea. There are several papers doing more or less the same, but if you analyze them carefully, you see that on average the accuracy is not that good, and that only high accuracies are obtained when the mutations are similar to the ones that they use to train their systems. In addition, these systems try to cover as many mutations as possible, so they tend to use very general features that lack the important features that are specific to a protein family of interest. So we propose here a system which is specific of a protein family of interest, in this case the kinases. The first step is to collect information on mutations and features specific to the protein kinase family, and we publish a number of papers on the collection of features from the kinases. Then the second step, which is the one I'm going to talk to you today, which is the analysis of the common features of the mutations. And there are two papers here, one already published and the other one submitted. And later on, the implementation of a server, which is the future work, and we have not done at the moment. So, which is a system which is capable of predicting the pathogenicity of any novel mutation we face after an experiment. So, we chose the protein kinase superfamily because, as you probably know, they are key proteins in the biology of cancer, but also because they are very modular and there are structural modules that are pretty well studied, and we know which function more or less they have, so this could help us to interpret the mutations. In the particular case of the ATP binding pocket, which is over here, we use two different definitions. In the first hand, we use the definition by FireDB, which is a server we have at our group, which provides the definition of the catalytic site as those residues that are in direct contact with ATP, in this case 32 residues for the kinases. And we use another more restrictive definition of catalytic site, which is the one provided by Knight and collaborators, where only 5 very conserved residues are taken into account. Regarding the mutations, we were able to collect two different kinds of mutations. On one hand, we have the pathogenic mutations, that are the mutations that are characterized as disease-associated in kinome mainly, and we got 130 pathogenic deviations in the protein kinase domain. And on the other hand, we have the neutral mutations, the SNPs from DB SNP. These are not related to disease. And we got 200 neutral mutations within the protein kinase domain. Okay. Our collaborators in the UCL stored all these mutations in a database and provided as well a structural analysis of these mutations. So they assessed whether the mutations were conserved, if they were reported to affect functionality, for instance, if they are in contact with another protein chain or a ligand, or whether the residue is annotated as functionally relevant in Swissplot or Tremble. In this database, there is also information about the stability change of the mutations. For instance, if there are classes or empty spaces, disruption of important bonds inside the protein, strange residues in positions that are supposed to be hydrophobic or hydrophilic in the core. So after analyzing all the features of the different classes of mutations, we found that neutral SNPs in the PK domain are likely to introduce empty spaces in the core of the protein. By contrast, pathogenic deviations, the ones associated to disease, in the PK domain are likely to disrupt the binding interfaces, both protein-protein interfaces or protein-ligand interfaces. These results are striking because the overall behavior of the pathogenic deviation is both affect stability as the protein kinase ones, but also to affect positions that are important for stability. So the take-home message of this analysis is that PDs within the PK domain are located in functionally relevant positions, but they are not in positions that are important for stability. In this, to enhance this study, we wanted not only to study the residues, but also the environment of the residues. So we considered the distances from the residues to the important features we want to analyze. For the particular case of the catalytic sites, we have here the catalytic site. In yellow, we can see the catalytic site as defined by FireDB. In bubbles, we highlight the residues that were also in the night definition of the catalytic site. In light red, we can see where the pathogenic mutations are. In dark red, we see those mutations that are both pathogenic and part of the catalytic site. In light blue, we see the neutral SNPs, and in dark blue, we see those that are part of the catalytic site, but also neutral SNPs. So we see that more or less pathogenic deviations allocate near the catalytic site. Although there are others that are outside the catalytic site, most of them are close to the catalytic site. If we plot the distances, we see, well, here is the distribution of distances in bins of 1 angstrom. See, is the closest distance from any mutation to any residue in the ATP binding pocket. So in red we have the pathogenic deviations, and in blue the neutral SNPs. As you see, in both definitions of the catalytic site, we see that there is a clear tendency for pathogenic deviations to be closer to the catalytic site than neutral SNPs. We repeated this kind of analysis in different structural features. For instance, we use the catalytic site from FireDB, catalytic site from the definition of Knight and collaborators. We also used the 3 determinants, which are those positions that are providing the subfamily specificity to the protein. Are those positions that are similar to the members of the subfamily, but different to members of other families. We also assess the accessibility, sequence conservation in 2 different definitions, and structural conservation. If you see the results, we see that all the features related to functionality show a similar tendency. Pathogenic deviations, the ones associated to disease, are closer than SNPs to these features. By contrast, when we study the stability-related features, we see that or there is no significant difference, or even the SNPs are closer than the pathogenic deviations. Okay. If we compare these results, which were performed using germline mutations, with other results we had in a previous paper using somatic mutations, driver and passenger mutations, we see very similar results. Driver mutations, which are the ones that are supposed to be in the development or involved in the development of cancer, are located near functional hotspots, whereas they skip regions important for stability. So the message of this comparison is that no matter the nature of the mutation, both germline or somatic, both behave similarly. Okay, conclusions. Pathogenic mutations present differential characteristics with respect to neutral mutations that can be used to predict the pathogenicity of a novel mutation. Despite their very different nature, somatic and germline mutations, pathogenic somatic and germline mutations are located in similar regions of the protein. Pathogenic mutations within the protein kinase domain display a preference toward certain residues that are those that are involved in ATP binding and specificity, whereas they try to skip other types of residues that are involved in accessibility or stability. And just to highlight that pathogenicity in kinases is different to pathogenicity in other protein families. So, let me explain you with an example why this is important for us. After analyzing the results we obtained, we saw that there was a mutation in the human insulin receptor that was an important hit in our results. It was this mutation over here, which is a shift from alanine to threonine in this position. And in our results, it was both in a catalytic site, but also in a T-determinant position. So our prediction is that this mutation is very likely to be pathogenic. We checked the literature, and we saw that this mutation is related with diabetes mellitus, which is resistant to endogenous administration of insulin. That also this mutation is associated with acanthosis nigricans, which is a defect in the skin that leads to a uncontrolled growth of cells in the skin, and also in women is associated with polycystic ovaries. So we are able to spot those mutations that are important using our features, but this is kind of trick because we have done this manually. The aim of our research is to do it automatically. So our aim is to develop a method that is capable of using existing knowledge from the literature or from genome studies or wherever to train the predictor so it learns how to classify the mutations based on the set of features we have analyzed. Then we evaluate the accuracy and coverage of the method and identify the most important features. And finally, we use this method to predict the pathogenicity of any novel mutation we face from whatever analysis we need to. Finally, I would like to thank all the people I've worked with in this project. Most of them from the UCL and all my group from the structural biocomputational unit. Many thanks.
Any questions?